The task ahead of us is to start to understand resistance mechanisms to RET gene fusions. There's been a little bit of data now published at meetings suggesting that solvent front mutations are going to be seen, and that leaves the door open for the development of new RET TKIs that can overcome that type of resistance for this. So hopefully having later line selective TKI options for patients who become resistant to pralsetinib or selpercatinib. The other, I think, exciting space that we're starting to see is we've observed, at least in a handful of cases, that RET gene fusions are actually resistance mechanisms to other targeted therapies. So we've seen RET gene fusions as resistance mechanisms to osimertinib in patients who are EGFR mutation positive, or patients with RET gene fusions who are resistant to alectinib, which is a tyrosine kinase inhibitor for an ALK. And there's already been some published data of combining drugs together to overcome this resistance. And I think, again, that's made much more possible by having selective drugs without a broad toxicity profile from hitting other kinases. So having selective RET inhibitors also allows us to combine these drugs better to overcome resistance in other disease settings. I think the data from emerging RET gene fusion inhibitors, these selective RET inhibitors, is really going to make a meaningful difference for this population of patients with RET gene fusions in the same way that it's made a significant difference for patients with lung cancer who have other oncogene targets in their cancers. And I think it just further emphasizes the need for broad testing in all of our patients. There's really no excuse now to be doing singleplex or sequential testing because there are so many potential targets for our patients and so many good drugs that we really want to be identifying the appropriate therapy for our patients, whether they're RET gene fusion positive or other oncogene positive patients, so that we're giving everyone the right therapy up front.
RET inhibitors will certainly add to the armamentarium of treatment options that we have for patients with non-small cell lung cancer. Although, yes, this is a rare population of people, it absolutely adds to a significant treatment option for the people who have this tumor. And I think what is most significant and remarkable in lung cancer over the course of the last decades is how we really have created this environment of precision oncology for our individual patients. And although, yes, we used to treat everybody with the same treatment, we now can really individualize treatment for the individual patient so that they have the best responses, the best survival, the best toxicity profile, so they can live for as long as they can with the best quality of life they can in the setting of an advanced illness.
I think that, first of all, it is very exciting that we have now potentially new drugs, rather, we have now new drugs that are going to be available for another group of lung cancer patients in a relatively short period of time. This is very exciting for our patients. It does make it a little more complicated for us because there's another gene alteration that we need to be aware of, and that we need to ensure that our patients' tumors have been tested for this alteration. Try and be sure that an appropriate test has been done. As I mentioned, that a DNA-based NGS assay may not identify all translocations. It is essential that we are aware of that information and that the lab that we are sending the tumors to does perform a RNA-based NGS assay. Finally, it is also important to note that in RET translocations, there can be multiple gene partners. With ALK, the primary, in over 80% of the patients, the gene partner is EML4. Whereas in RET, there are multiple gene partners. Now, with multi-kinase, tyrosine kinase inhibitors, we found that the drugs may not be as efficacious with each of the gene partners in RET translocated non-small cell lung cancer. Early data suggests that differential efficacy is not seen. That is, RET-specific inhibitors appear to be beneficial in all RET translocated non-small cell lung cancer, but this data needs to be further analyzed and try to assess whether that remains to be true. In addition, we are starting to see progression in patients treated with RET-specific inhibitors. And we're starting to learn the mechanisms of resistance that leads to patients progressing on these drugs. And based on that understanding, there are now novel RET inhibitors that are coming into clinical trials. And so one needs to now become familiar with these drugs as well, so that patients who are treated with RET-specific tyrosine kinase inhibitors, who, when they do progress, we make our patients aware of these trials with these novel drugs, which may land up providing them the most clinical benefit, in addition to the other treatment options that we already have.